This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, Episode 10, December 3rd, 2008. I'm Vincent Racaniello.
I'm Dick Despommier.
And today we have a guest, Alan Dove. Welcome, Alan.
Thank you. Good to be on the show.
Welcome to TWiV, the podcast about all things viruses. Now, Alan, you are a science journalist. Is that correct?
That's what they tell me, and the checks have been clearing, so yes.
And you also have some experience in virology, isn't that right?
Yes, I do. As a matter of fact, I was in Vince's lab and got my PhD in this field.
Awesome.
I'd forgotten.
Yes.
You know, he's sitting here smiling like a Cheshire cat. He's very proud of you, Alan.
Yeah, I am. Of course I am. I'm very happy you've joined us, and I have to tell you all that Alan is way ahead of the curve. He wrote the first website for my laboratory.
Wow.
Years ago. How many years was that, Alan?
That would have been about 1995, early 96, somewhere in there, when I should have been purifying poliovirus receptor.
That's right. But he was ahead, and he made the first website, and we still have remnants of his work on our website. We'll put some links for you to see later. So today we have, as usual, a collection of very interesting things to talk about. We're in my office because Alan is joining us by Skype, and since Dick is a bit of a Luddite technologically, we've come here with all my equipment. So we're going to hear weird noises today. We're going to hear sirens on the street. We're going to hear the cold room compressor, which is just above me. And we may also hear air, but hopefully I can remove it in post. So let's get going. First thing on the agenda, turn on the recorder. I've got that done. Now, we had a couple of emails from last time I want to just go through. Chris from Canada wrote, our last episode was about Lassa. Do you remember, Dick?
I do.
He wrote, I hadn't heard about bringing the Lassa-infected patient into the USA by regular plane. Now we can't even pour sodium chloride down the sink. Things have changed, haven't they?
Things have changed a lot.
The regulations are incredible.
Yep. Well, since they didn't know what she had, they didn't know what to take precautions for.
But nowadays they would take precautions without a doubt.
Absolutely.
Chris also added, we talked about using immune globulin, remember, to treat infections. And he added that it's also used when you get a vaccinia immunization. They have some on hand in case things go wrong.
Right.
And you get a generalized vaccinia infection.
Right.
So that's a good thing to add.
It is.
Now, Stephen from the UK wrote, I ordered a copy of Fever from Amazon. Managed to get a second-hand copy, browned pages and all. The copy I got was published in 1977. This physical copy is older than I am.
Cool.
It's an old book. That's great. I'm glad that he went out and got it. I hope you enjoy it, Stephen. Jeffrey from New York. I just wanted to write to you to say that I think you guys are doing a great job with TWiV. I've been looking for a good science podcast for some time, and now I've found it. Your netcast has quickly become one of my top three alongside Twit and Dignation. I don't know if you know Dignation, Dick. Very famous pod. Alan, you must.
Yeah. That puts you in the big leagues there.
Of course, Dignation makes money.
Oh,
and we don't. We don't make any money.
No.
Now, Stephen, Jeffrey also pointed out a website which I didn't know about. Maybe you do. It's called healthmap.org.
I've heard of
it. I had not come across this, but I was absolutely astonished.
It's fabulous.
Really, really impressed with the implementation.
It's immediate, right?
It is immediate.
It's basically a world map, and you can select what data feeds you want, like ProMed, WHO, Google. You can select the disease. You can select the country. I don't know how up-to-date it is. It's not real-time, obviously, but it's very good. And there are little pins all over where there's an outbreak of something. So we'll put that link in the show notes. That is really nice. Thanks a lot, Jeffrey. So our readers are getting involved. This is great, or I should say our listeners. That's really good. Thanks, all of you, for the comments. I wanted to mention one thing. before we move into some stories, and that is there is a new website, relatively new website called socialmedian.com. And this is a website where you can basically take, if you're at another website and you see a newspaper article or any kind of article of interest, you can highlight the first few sentences. You click something on the menu, and it gets put onto this website. And then other people can look at this and clip it if they like it. And you can make groups of information. It's sort of like Delicious, but it's more socially oriented. So I have made a virology network at socialmedian.com.
Neat.
And I'm every day I look at virus news, and now when I come across an article, I clip it to Social Medium, so they're all there stored. And in fact, our readers, if you find things, you can join this network and add articles, and we'll talk about them if we so choose. So socialmedian.com. Have you heard about that, Alan?
I hadn't come across it, again, until you set out this agenda, but looks very cool.
Well, I guess you're learning things already, Alan.
Yeah, I'm already, the first episode of TWiV that I listened to a few weeks ago, I learned some things.
Great. That's what we hope everyone's doing. Okay, now the first story up is in two parts, actually. The first is an article in PNAS, the Proceedings of the National Academy of Sciences, about SARS virus. And then the second part is how it was reported in Wired Science, which is an online blog, I guess, or some reporting thing. The original article is a bit interesting. It's called Synthetic Recombinant Bat SARS-like Coronavirus is Infectious in Cultured Cells and in Mice. So what they basically did here was that we have sequences from a number of bat coronaviruses, which are thought to be the precursor of SARS, right?
Mm-hmm. Right.
But none of these viruses grow in cells. No one can get them to grow. So what they did in this paper was to assemble the DNA, the whole genome of these viruses as a DNA copy by synthesizing oligonucleotides and piecing them together. And then they assemble
synthetic virus.
Sort of, but it's based on a known sequence that came out of the bat. But it is a synthetic, and it's a synthetic recombinant bat SARS-like coronavirus. And then this virus does not grow. It doesn't produce. If you put that DNA into cells, you don't get viruses produced. It will grow. It will produce RNA in the cell, but it doesn't make infectious virus. So what they did was they added the spike glycoprotein of a human SARS virus to this bat virus, and now the virus will grow in cells and in mice. So, I mean, either there's a problem with the sequence of the bat glycoprotein, or maybe it just doesn't grow in the cells that they tried. But in any case, you now have a virus, which is mostly bat coronavirus, and now you can grow and do experiments. So that's the observation. It's interesting. If you're interested in SARS and you want to work on it, obviously this is very good because you can work on these bat viruses. Now, the problem I have here is the Wired coverage. And have you seen this before, Alan?
I saw the headline of this. I have about a bazillion things in my RSS feeds that are things like this, and I saw the headline. I thought, oh, that looks kind of interesting. I skimmed maybe the first paragraph of the story and thought, this is the type of hype that comes out when anybody mentions synthetic biology these days. So I kind of dismissed it.
Well, I probably shouldn't dwell on it too long, but I just want to point out the name of this article. We'll put the link in the notes. It's called Synthetic Viruses Could Explain Animal to Human Jumps. And the first paragraph, in a technical tour de force with potentially profound implications for the study of emerging diseases, researchers have built the largest ever self-replicating organism from scratch.
Yes.
I guess you're right, Alan, that you want to make your article interesting, right? So this is... In journalism school, you always tell people that you want the lead to really grab the reader, and so they're trying to put this into a context where people will say, oh, wow, this is huge. Yeah. So I guess I didn't read it from that viewpoint. I'm reading it from a scientific viewpoint. And it said, a few paragraphs down, it said, just a decade ago, artificially constructed viruses seemed like science fiction. Now, this really bothers me.
I bet.
Right? Because, in fact, the first virus made from DNA was a bacteriophage by Charles Weissman in the 70s.
We made tobacco mosaic virus, too.
Tobacco mosaic virus. Long time ago.
And back eons and eons ago, I think some kid in David Baltimore's lab put together a cDNA copy of poliovirus, right?
That's right. And that, that was the first animal virus. When put into cells, led to the production of virus.
Right.
Now, the difference here is simply that they put together the DNAs by purchasing oligonucleotides and then putting them together. And this was actually so. What they refer to in this article a decade ago is a paper in Science by Eckhard Wimmer and his colleagues, where they put together the whole polio DNA copy from oligonucleotides that they purchased and showed it was infectious.
Cloning by phone.
Yeah, you remember that, Alan? That got huge publicity, right? And it was in Science, it was in the Times, he was on TV. And I wrote a letter to ASM News saying, this is known for already 15 years.
Exactly.
DNA is DNA, isn't it? It doesn't matter if you buy it or if you...
Right.
So,
as long as it's not degraded.
If it's not degraded, right. So I have a little issue with making a big deal of this because DNA is DNA. So I wrote a comment on this website, get your research down. Don't say 10 years because it's a little bit more than that.
Right.
So, Alan, am I overreacting to this? Or?
Well, the thing that struck me going back to this article, as I say, I see stuff like this and worse in my RSS feeds all the time.
Sure.
Then I went back to this and looked at it through that lens, and I thought, yeah, this is pretty seriously overblown. But the thing that really grabbed me the second time around was the depth of the ignorance in a lot of the comments.
Yes.
And now you say you commented on it, but if you just look at like the first 20 comments that came up, these people who are talking about, well, this is evil research because we're going to have engineered SARS breaking out and killing everybody, and I had thought that the readers of Wired were a little bit more. What's the word I'm looking for here?
Scientifically sophisticated?
Yes, thank you. That's diplomatically put. So there's this whole atmosphere of, oh, gee, this is frightening, scary technology, when in fact, I would expect, just from a purely evolutionary standpoint, a synthetic virus is probably going to be at a severe disadvantage if it gets out.
Absolutely.
Well, I hate to say this, but they've already reconstructed the influenza virus that caused the 1918 outbreak. So if you want to worry about something, there's something you can worry about.
That's one to worry about. Well, I mean, we should point out that in this case, they've just made a virus that already exists.
Right.
That's been produced by evolution. What are we to say that we could create a virus from scratch? Zero. We could not do it. Alan, you're absolutely right. We couldn't make a killer virus if we wanted to.
Well, if there were some virus that were capable of infecting all of humanity and wiping us out, then we wouldn't be here having this conversation now.
Right. But I think there is a virus that's close to that. It was the HIV-1, right?
Right.
It's getting very close. Which is our next subject, actually. But before we get to that, this whole business with the SARS and the bat virus is very interesting. I don't think we've talked about it very much. At some point, we should talk about the ecology of that.
Right. We did talk about bats, though. I remember that because we talked about rabies.
A little bit, yeah.
I remember we said 1/3 of all the mammalian species out there are bats.
In fact, in this month's Microbe, there is an article on bats as a great reservoir of viruses. So we have to revisit that sometime. The whole idea, all human viruses came from animals at one point or another. Wouldn't you guys agree?
We would.
Definitely,
right.
Yeah.
I agree. So, and as we said last time, the Lhasa incident was the first time we realized that these come out frequently and they can make you sick.
That's right.
I was at a lecture by Ian Lipkin this week, and he said something. He quoted Steve Morse, who we should have on sometime also. 50,000 vertebrate species. Is that about right, Dick? You'd agree?
With Ian, I would agree. I have no basis for disagreement.
Well, that's a quote from Steve Morse. So you agree with Steve Morse?
Well, I co-teach the course Emerging Infections, and far be it from me to disagree with my coworker.
So if there are just 20 viruses per species, that's 1 million vertebrate viruses, which basically is more than 99% remain to be discovered still.
Correct.
A lot of viruses out there.
Exactly right.
So this paper on SARS emphasizes the fact that you can't grow a lot of them, but maybe if you could make the DNA and put it together, you could do something in the laboratory. And I think that's fine. As you know, a lot of the viruses that Ian Lipkin identifies can't be grown.
Right.
So maybe this is a good approach.
Sure.
Okay. This week, of course, on Monday was World AIDS Day.
Yep.
And there was a lot of hype about an article that was published in Lancet, and for which there was also an editorial in the New York Times. I don't know if you saw that. Either of you saw that. A plan to eradicate AIDS. And the idea here is that you find out everyone who is infected with AIDS, and then you start treating them with antiretrovirals immediately. So right now we wait for people to be symptomatic, right? And then you treat them, and it's tough, especially if their CD4 counts are very low already. But in this case, they would go in particularly regions of Africa and other third world countries where there's a lot of infection, do massive surveys.
Using PCR, I presume.
Whatever they want to use, and say, okay, you people are infected, we're going to start treating you right away. And they may not even be symptomatic. And then the Lancet article was actually a mathematical modeling of why this might work or not. So what do you guys think about that?
The same thing I think about when I remember how chloroquine resistance arose to malaria, both in Colombia, South America, and also in Southeast Asia. In Colombia, South America, they thought that chloroquine was such a panacea that they started to put it into the salt. That meant everyone received chloroquine of some level. And what it did was it selected for R1 resistance in the malaria parasites. Then they had a tougher time treating them. That parasite took hold, and the next thing you know, you had R2 and R3 resistance, and now chloroquine doesn't work there at all. The same thing happened in Southeast Asia, but in a different way, because that's when we had the Vietnam War going on, and virtually everybody was treated with chloroquine. And no one liked it, so the ground became green with these pills that people were discarding because it made them sick. And I guess they would rather be fired at by the enemy than take these pills and feel miserable. As a result, they took some treatment, but they didn't take it all the time.
Right, sure.
And they got another R1 resistance, and then the next thing you know, they've got total resistance there too. So when you start mass treatment with a drug, even though there are 3 drugs involved in this, it's just a matter of time before the virus leapfrogs over all of those barriers and becomes now the resistant organism that takes over the world again.
And we know we get resistance to the triple treatment anyway, even in developed countries.
It has to happen. So it's just a matter of time as to when that happens. So I guess the thinking here is that the faster you act and the more people you treat, the less likely it is that the mutant will arise before you've eradicated the disease. So it's a race against time.
It is, yeah. But your point is well taken. You have to be vigilant.
Well, it's interesting because when I saw this, I also thought of malaria. And what came to my mind, anytime somebody talks about disease eradication, of course, they're still trying to eradicate polio, but there was a little-known or little-remembered effort, perhaps, in the 1950s through the 1970s to eradicate malaria.
Yep.
And I'm sure Dick remembers this.
Oh, I do.
and it was... It ended up being an utter disaster. The approach was a big part of the approach was to spray DDT all over the place to wipe out the mosquitoes. The mosquitoes became resistant. The DDT, of course, we now know is still in the environment because it's persistent. It doesn't go away. And what we're left with is plenty of malaria. And we've still got the problem, and yet we've got all these additional problems too. So eradication approaches, I'm immediately skeptical. But I will say, I think there is a lot of merit in these types of mathematical models because what they show is what's theoretically possible. Now, there's a huge gulf between theory and practice, particularly with a disease like HIV. But it's important to have something like this that shows, look, assuming no change in technology, assuming current technology, it is theoretically possible to control this virus. Now that, as I say, that sets one boundary that says, if you were able and willing to take these very extreme measures, and I think a lot of what was lost in the coverage of this was that what they're advocating it would be massive. Universal voluntary HIV testing for everybody in Africa is going to be a logistical nightmare. Yeah. But in theory, it is possible to do this, and there's no scientific reason to believe that it couldn't be done. There are lots of social and policy-making reasons to believe that it couldn't be done. So I think it sets an important boundary for the debate to say, okay, it's possible. Now, do you want to work toward that goal, or do you want to take a different approach and try to control the disease rather than eradicate it? But the upshot is, I think it's an interesting study. I think maybe some of the discussion of it was a little bit off, but it is important to have these types of conversations.
If you compare and contrast HIV control with smallpox, for instance, which is that other virus that we claim to have gotten rid of, the reason why we could get rid of smallpox is because there were no reservoir hosts, and we had a good vaccine. And we had a vigilance with regards to finding out where these cases were, because all you had to do was look at somebody to find the smallpox scars to find out where smallpox still resided. Whereas HIV is a silent killer. It takes a long time to come up. The incubation period is months to years. It's a very, very difficult situation.
No, that's why... Sorry, go ahead.
No, what I was just trying to say was if you could identify hotspots that served as dissemination centers for HIV infection, like inner cities of maybe West Africa and maybe South Africa, where large numbers of people come, they exchange bodily fluids, however that occurs. Maybe those are the places where this approach might do the most good.
And highways and trucking routes.
Yeah, exactly right. But again, the big danger is that you're going to select for resistance eventually.
Right.
And that's then you're just emptying the gunpowder from your bullets and trying to fire them, and they just don't work.
Well, I do agree. I think it's good to get the dialogue started, and because obviously we can control the infection better than we're doing it.
That's right.
I mean, it's just a matter of getting diagnosis and antiretrovirals to Africa.
Yeah,
right.
But with some notable exceptions, like the country of Senegal, for instance, has a very low rate of HIV, and that's because the family unit is very stable. They're very devout religious people. They don't share sexual partners. They have a different ethic and an ethos. And whenever you have that as a dominant theme, you don't have AIDS.
Right. Well, but there's also, in Senegal, isn't there also a very high rate of HIV-2 infection?
I don't know that.
Yeah.
Okay.
That may well be, yeah. We'll check on that.
But, okay.
Because,
Gentlemen,
do you want to leave, Dick?
I'm afraid that I'm going to have to bid adieu.
I won't shed any tears.
I hope.
Will you come back next week, Dick?
Next week I'll be in India.
The whole week?
I'm being in India until January 6th. I'm on a trek. I was invited for 2 lectures starting next week, and a lecture in Bangalore and then in Pune. Mm-hmm. And you can't go to India without seeing the Taj Mahal.
Yeah, excellent.
So,
sounds
unfortunately,
great.
I'm not back for TWiV
All right.
13.
It will be Alan. Maybe you can join me for the next weeks.
That sounds good.
And we'll pull in some other people, because we need more than one person to replace Dick, don't we?
Oh, absolutely.
No one's listening to that one, I hope.
Well, have a good trip.
Thanks, Vince.
And what are you doing? You're going to teach a class now, right?
I'm going to go teach a class right now to second graders at a school that's associated with Columbia University, and I'm thrilled to be asked to do this. I'm going to read them 2 books. One is called Old MacDonald Had an Apartment House, and the other one is called The Lorax. And we're going to try to get these kids to start thinking ecologically at a young age.
Tell them to start listening to TWiV.
I will.
Please.
I will.
We need to get the ratings up.
You know what they're going to do? They're going to tell their parents about this, and the parents will not let them listen. Because
but they probably have the iPods, the kids, though. All right, Dick. Good to have you anyway.
Alan, very nice to have you on the show.
Good talking to you, Dick.
Yeah, same here. We Alan and I shared a common experience down in Mexico City about a month and a half or 2 months ago, and you did a marvelous job of writing that up, and really appreciate the work that you put into that.
Oh, glad you liked it. Good times.
He had good training.
He had wonderful training.
I'm just kidding. No, not my doing. All right, Dick. Close the door behind you, please.
I shall. I'll turn the light off.
All right, you're staying there, right, Alan?
I'm still here.
Good.
Okay.
We'll continue.
Bid you adieu.
Bye. No problem. And enjoy India. No, it's okay. I'm glad we got Alan now, then. I can't do this myself. Nobody wants to listen to me. Bye, Dick. Alan and I will go forward, and then we'll get a couple of other people. We'll get some various guests, you and I, Alan, so it'll be fun.
Sounds good.
Okay, one more news story here. I've seen this one on and off for quite a while now. Herpes virus killing elephants in zoos. Have you heard about this, Alan?
Yeah, I did hear about that. Kind of what you're saying. It's come across my radar a couple of times, and I kept thinking, gee, that's odd.
Yeah, so I found an article at the beginning of November. There was a death in a Calgary zoo.
Right.
And then just this week, there were a couple of others. I'll put the link to the article in the show notes. But it turns out these are kind of different herpes viruses. They are gamma herpes viruses, first of all, which means they're like cytomegalovirus. They're not herpes simplex viruses.
Right.
And they seem to be, let me see if I can get this right. Now, there are two kinds of elephants, right, Alan? They're Asian and African elephants.
Asian and Africans.
And I think this virus is pathogenic for Asian elephants.
Right.
But not for African.
Right. The African elephants seem to get a very mild few sores here and there, and then they're fine.
Right. But particularly the Asian babies get this. So they seem to get it from their mother at birth, right?
That's the theory. Although I was just, kind of like when I used to prepare for classes in graduate school, I was just looking into this topic right before we got on, and it seems that there's been some analysis, some phylogenetic analysis, where they sequence the genes of the different isolates of the viruses from a bunch of different cases of this. And they found, I think they sampled 18 elephants, and they found 18 different distinct strains of the virus, suggesting that each one came from a different source. So it's not quite clear what the etiology is, where they're getting this disease, except that it's possible that you have a lot of zoos would have Asian elephants and African elephants, and it may be that the African elephants are acting as carriers, transmitting it to the Asian elephants, and maybe it's passing that way.
Well, I suppose at some point you get new elephants from the wild, right, and then they could have the virus. So these, the older elephants probably have it latently in their nerve cells or wherever, as do other herpes viruses.
Right.
And then at birth they transmit them, or even maybe at the young stage of the calves, the parents might have a lesion which they rub against the baby elephant and they infect them as well.
Sure.
And for some reason in the babies it's lethal.
90% mortality. Yeah.
And I guess an elephant in a zoo is a big deal. It's probably not a cheap animal, right?
Well, no, and one of the things I thought of when I was reading this was they were talking about this zookeeper who's studying this condition, and I thought, wow, that would be a tough model organism to work on.
Yeah, they're huge, and people do like the elephants, so you don't want to lose them. But I read one article which said basically they're not going to have any more Asians because all the babies die.
Right.
And I've read in one zoo, every baby that's been born in the last few years has died because of this infection.
Right.
So it is an issue. We should point out that the virus appears to cause hemorrhaging. It infects endothelial cells, particularly in the heart. And the capillaries in the vessels rupture, basically, because they're infected with the virus. And so it's a bit different from other kinds of human herpes virus infections, let's say.
Right.
What would you do? Now, the other interesting point about this elephant herpes virus is that they have a TK gene. And this is different. The beta herpes viruses don't have TK genes. It's something that's a property of the alphas, the herpes simplex viruses. So that means you could, in theory, treat these infections with acyclovir, which requires a TK to be specific for alpha herpes viruses, and now in this elephant herpes virus.
Right, and well, in fact, when I was looking around for more information on this, there are I think a half dozen of these where they have actually rapidly, they got the test result, they found the elephant was positive for this herpes virus, and treated it immediately with famciclovir. Mm-hmm. And in several of those cases, they actually managed to keep the elephant alive.
Mm-hmm. Yeah, I guess
so that it does appear to be effective. The problem is that apparently when they do the PCR test, the blood test on these elephants, they've never had a healthy elephant come up positive on the test. Mm-hmm. So the virus is presumably lying latent, probably in a ganglion somewhere in the nervous system, and is apparently not. There's just no DNA of it circulating in the blood. And then by the time it reactivates, this is a very, very fast developing disease. In many cases, 24 hours, the elephant's dead. But apparently, if they detect it and get to it fast enough and treat it, it is potentially treatable. And with famciclovir, which would be consistent with what you're suggesting.
Right. So in this case, you have to either develop a different way of looking for the virus other than a blood test, which would be hard, I guess.
Right.
Or be very vigilant and work quickly.
Right.
I can imagine it would be difficult to figure out what dose to give an elephant of these anti-herpes viral drugs, right?
Yeah, I guess you'd have to do it by body weight, which... Yeah.
We should probably point out that these drugs we're talking about, the acyclovir, which was the original, and all its derivatives, like famciclovir, are all chain terminators. And the way they work is they have to first, one phosphate has to be added to these compounds by the viral enzyme TK thymidine kinase. So these compounds get into cells. They're not phosphorylated initially. They get into cells, they get into all cells, but the ones that are infected with herpes viruses have TK gene product in them. They phosphorylate this drug, and then cellular kinases add a second and third phosphate, at which point they can be used by DNA polymerase. And at that point, when they're incorporated, they can't be extended anymore, and the chain terminates. So they have low toxicity because they are only active in viral infected cells.
Right.
So this is good for the elephants that these viruses happen to have a TK gene. Otherwise, it would be difficult. There are some antiviral programs for cytomegaloviruses in various companies. One of them is targeting the viral protease, which is a unique enzyme. It has an unusual fold, and therefore, in theory, you could make specific drugs that don't have much toxicity. So maybe those could be useful. But it's this is just another example of all the viruses that are out there. Perhaps because we have more zoos now with more exotic animals than we ever did, this is becoming a problem which we never saw before. But I'm sure there are many more viruses and all kinds of animals that are going to raise their heads in the near future.
Well, also, I gather that there's the breeding programs have gotten more serious since the Asian elephant is now endangered in the wild.
Right, exactly. You can no longer import them, right?
Right. So the zoos are trying to step up and replenish the population, and right when they start doing that, they discover they've got this virus problem.
A good point. I wonder if a similar issue is preventing pandas from reproducing effectively in zoos, at least in American zoos, right?
It's a good question. Yeah, I know. Well, in zoos all over the place, the Chinese have had some luck with it, but even there, it's a very, very difficult process.
In general, our understanding of viral infections of non-human animals is pretty rudimentary. As this,
to put it mildly,
because there's limited money, and where there is money, it goes into investigating human diseases. But there are plenty of animal diseases. What come to mind right now besides this elephant one are the various encephalitis infections of horses.
Right.
Which are economically huge. So there's a lot of attention given. There are even vaccines for these viral infections for horses specifically, equine encephalitis virus, because it's an huge economic issue. But there are many others for which there isn't an impetus, which is too bad because I think we could learn a lot. I personally think we should be looking at viruses in animals much, much more than we do.
Well, especially since that's probably the source of most novel human outbreaks. If you want to keep track of where the next major disease is coming from, having some kind of a tracking program in animals would be a good start.
Absolutely. Yeah, I mean, we have the technology now to track very easily what is circulating in animals, and it's just a matter of setting up programs to do that. But it's very difficult to get funding for these kinds of studies, as you know, because they're not what we call hypothesis-driven studies, which is what many of the funding agencies like to fund.
Right.
But they're certainly worth doing, in my opinion. Oh, well. So much for lamenting for today. Well, I think we ought to wrap this up. Let's do a few things in closing here. And we started a few weeks ago naming a science podcast of the week. And you should think about these for the future, Alan. Maybe you have some that you like. But today I picked one called Futures in Biotech. Have you ever heard of that one?
I have. I haven't subscribed to it, though.
It is done by Mark Pelletier. And the podcast, basically, he speaks with various scientists about very specific topics. It's beyond virology. It's molecular biology and other fields. So it's about an hour interview. The last one I listened to was with Cynthia Kenyon on telomeres.
Okay.
They're well done. They're informative and very broad-ranging. This podcast is a member of the TWiT network, TWiT, This Week in Tech.
Right.
And, of course, we are TWiV, This Week in Virology, and they're TWiT. And there are many other podcasts, mostly dealing with technology. And they're all very good. But we'll put a link to that in the show notes. It's a good podcast. Then one of our readers, I haven't got this in the agenda, Alan, but one of our readers suggested we do a science book of the week as well, because we've actually mentioned some science books over the past few weeks, and I put the links in when we do. So he said, why don't you do that? Because we're always interested in hearing what's good to read. So, for example, last week, Fever. No one had really heard of Fever. Had you heard of that book, by the way?
I had heard of it, and I have not gotten around to reading it.
Yeah, so as I had talked about, it was really inspiring for me way back when. So anyway, keep that in mind, Alan. Today I thought I would pick a little virology textbook, which I enjoy very much, by Alan Kahn, and it's called Principles of Molecular Virology. Alan Kahn, of course, has his own podcast and blog called Microbiology Bites, which we picked a couple of weeks ago. And this textbook is excellent. It's a very simple, small textbook on the basics of virology. Really good for undergrads, but anyone who wants to get a really rudimentary understanding of virology. And we'll put a link to that in the notes. Of course, we have a little virology bookstore growing over at Amazon. And every time we mention one of these, we put the book in there. Have you heard of that book, Alan?
I haven't.
Okay, there's another thing you've learned today.
Yes, exactly.
So we will keep doing this in our future podcasts. So I want to thank Jeff for sending in his lists of podcasts as well. Futures in Biotech came from that list. So thanks, Jeff. And you people who are listening, spread the word. Get others to listen and have them sign up. Alan, where can people find you online?
Well, online, I'm pretty easy to locate. I'm now the second Alan Dove who comes up if you do a Google search. But an easier way to find me is at dovedocs.com. That's DOVDOX.com.
Very good. Alan Dove, thanks for joining us today, and you'll come back next week?
Absolutely.
Okay, well, we'll arrange that. So all of you, thank you for listening to us. We hope you subscribe. Send in your questions. Send in what you want us to talk about. Send in your news articles to social media and the Virology Network. We'll take a look at those and we'll talk about them. You've been listening to This Week in Virology. Thanks for joining us. See you next time.